get started. Hi everyone, and welcome to this Qlucore webinar on regarding Qlucore Insights in this case, or maybe Qlucore Diagnostics as well. My name is Fredrik Hellborg, and I'm Director of Diagnostic Sales here at Qlucore. And the title of today's webinar is Qlucore Insights: How to Set Up Your Bioinformatics Pipeline. And to explain all that, I have my colleague Caroline with me today. She will let you know exactly how to do that. Come to the questions when appropriate. And just an overview of the tests that are available for Qlucore Insights and Qlucore Diagnostics at the moment. So for Qlucore Insights, we have the BCP ALL test, acute lymphoblastic leukemia test, and it's also available in Qlucore Diagnostics. Then we have a lung model, or a lung test, I should say, and a bladder test, and an acute myeloblastic leukemia test. So these are the tests that are available as of today. And to the right, you can also see that some of the hospitals that are using these tests at the moment.
Today, I'm going to walk you through how to run sequence alignment and gene fusion calling for Qlucore Insights. And I use our BCP ALL classifier as a model. So this is a schematic overview of the bioinformatics pipeline that we have set up. It uses tools that are open source, but that usually runs on large Linux clusters. And this can be quite challenging for people that don't have a bioinformatics background. So we have simplified it a little bit. But just to show you very briefly the overview, we start with raw sequencing reads, pair-end reads from, for example, an Illumina instrument. And we align those reads to a reference genome using STAR. The output from STAR is an aligned biome file and a file called chimeric out junction, which collects all the chimerically aligned reads. And this file is then used for fusion calling. And we have three tools. STAR Fusion, which uses this chimeric out junction for calling fusions. And then we also run Ariba and Fusion Catcher. And these tools run directly on the FASTQ files. Ariba also runs STAR for sequence alignment. And once the pipeline is run, we collect the output files, the BAM file, and the output files from the three fusion callers as input to our Qlucore Insights model for a subtype classification and gene fusion detection. I will go more into details. But as I said, it can be challenging to run these tools. So we have set up a pipeline. We have a few requirements. First of all, you need a 64-bit Linux, Mac, or Windows operating system. And since alignment is quite heavy on memory and also on disk space, we require at least 64 gigabytes of RAM and 256 gigabytes of free disk space to run this. We have set up this pipeline using the Docker desktop or Docker containers. So you need a Docker desktop app. And you can see that we have pre-installed these bioinformatics tools in a Docker image. And you can see which versions we have installed. So these tools are ready to run directly in Docker. Of course, you need our Qlucore Docker image. And the pipeline also uses reference genome libraries. And these are actually downloaded by the Docker script. So you don't have to do this in advance. And we keep track that the correct versions are downloaded. And this only needs to be done once. It needs some disk space, and it can take quite a while. But since it's only done once, it should be okay. And the pipeline is run with human reference genome 37. The pipeline assumes a certain directory structure and file structure for the alignment. And I've exemplified this with different colors of these folders. So in green, we have existing folders that should be on your system, where we have data, FASTQ folder, and then the FASTQ files are organized in one folder per sample or case. And then the blue folders are created by the first Docker script, which sets up all the genome references. So it creates a reference folder and populates it with the downloaded reference files for each tool. So Star and Star Fusion uses the same genome reference file from CTAT, and Ariba and Fusion Catcher has their own. And then the second script runs the purple folders, or creates the purple folders, and it's the output folder and files that's created by the pipeline. So we have an output folder and then a folder for each sample or case where all the output, the BAM files and the Fusion files goes to. So to run the Qlucore pipeline, you need the Docker desktop app. You can go to the Docker website and follow instructions for how to install Docker on your operating system. Then you simply open the Docker desktop app, and it looks like this. And then you navigate to the terminal, which is in the lower right corner. And this opens a terminal within Docker desktop. And it's a command line prompt that is used to run the pipeline. So within the terminal, you load our Docker image from Qlucore like this. Simply copy paste into the terminal and hit enter, but you should change the path so it's suitable for where you have downloaded or saved the Docker image. And then you start by running the script for downloading the reference file. And you do this by running the Docker container and then the bash script, which is called setup reference sh. And I will go more into details for exactly what these lines of arguments are doing in the next slide. And then you run the alignment and fusion calling by these lines. And our script is called align and call.sh. And of course, change path to fit your system. But here are a more detailed view of what's going on when we run these scripts. As I said, you should start the Docker container. In this line, you can see that the path to the mount point, which is before the semicolon, is the path to where the data is on your system. And for me, this would be in this line called users, my initials, documents, projects, BCP, ALL, and data. And this data folder is then mounted in the Docker container. And after the semicolon, it should be called data. And this Qlucore Align Fusion's GRC837 is the Qlucore Docker image that we've loaded into Docker. And after that, we run the setup script using bash. And it only needs one argument, which is the data folder. And when it's run, it creates the subfolder under data called reference. And there it downloads the reference genomes and populates these three subdirectories. One for each tool, where Star and Star Fusion uses the same from CTAT, and Ariba and Fusion Catcher have their own. And if you want to see a full list of options for this script, you can just run with the help flag. and change the mounting point in the first part to fit the data on your system. And you should also use absolute path for your mount point, as I showed in my example for where my files are located. And then you're ready when all the reference files are installed and downloaded. You're ready to run the alignment and fusion calling with these lines. Again, if you haven't already, you should start the Docker container as we did in the previous slide, and then run the bash script align and call. And it requires 3 arguments. The first and second points to the FASTQ files within the Docker container, which is located in the folder data, FASTQ case 1, and then we have the first, the read 1 of the paired end reads, and the second argument is the read 2 of the FASTQ. And then we need an output directory, which points to the folder that collects all the output from the pipeline. So it's located under data, creates output, and then one folder for each sample or case. And if you want to see a full list of options for the script, you run it with the help flag instead. And again, of course, change the mount point to fit your system. And this slide just shows the full list of options that are available via the help function for the align and call script. It shows you how to run the alignment, how the usage of the script, that you need the first FASTQ file and the second FASTQ file and the output directory. But it also shows you that you have further options. For example, if you need to change the number of available cores on your computer, you can do this by the C flag or the cores flag. If you don't set it, it's defaulting to 4 cores. But what is actually happening underneath the hood of the Docker container when you run the script? And this slide just shows you the commands we use for running star alignment. It is all the arguments or commands are recommended by Star Fusion for downstream fusion calling. We've made one critical addition, which is the flag out filter multimap n max, which we set to 200 to handle genes in repetitive regions. And as you can see in this script, the first argument specifies the reference genome index files that we've downloaded. And the second argument specifies the FASTQ files, the read 1 and read 2. And then the third argument sets the available cores, which can be changed to what's appropriate for your system by setting this cores flag in our script. And then the output, the last argument is just that the output is sent to the output folder that we've set up with the script. And then when we've run star, we run Star Fusion. It uses the same reference library as star. Also here you can change the available cores. And then, as I said, Star Fusion uses the chimeric out junction file from star alignment as an input. And this file collects all the chimerically aligned reads that star detected. And then we add a flag called examine coding effects, or coding effect, which predicts if a fusion is in frame or not. This is done automatically by the other callers, but here we need to set a flag to get Star Fusion to output this. And this can be used as a filter in Qlucore Insights. if we only want to look at fusions that are predicted to be in frame, for example. And then the output is sent to our out directory folder. Ariba also runs star alignment and pipes the output directly to call fusions. So it's very fast because it can start calling fusions at the same time as star is aligning. We run star with the arguments that are recommended by Ariba. And during Ariba calling, no BAM file is created. Instead, it collects all the fusions in the fusions.tsv file that we use. And finally, we run Fusion Catcher with default settings. And here it's important to point out that the input folder, or the input in Fusion Catcher, is a folder, and all FASTQ files within that folder are aligned and called. So if a folder contains more for FASTQ files for more than one sample, the results for all samples will be in one resulting Fusion file. Therefore, we set up the folder system so that each sample has its own folder with FASTQ files in our pipeline. So, which files are used as input to Qlucore Insights? From the star alignment, we use the BAM file, which is sorted by coordinates. And star fusion uses the fusion file ending with coding effects, because then we have the information whether or not a fusion is in frame. Ariba, we use the fusions.tsv. And for Fusion Catcher, it's important to use the output file that ends with HG19 as input. And these files are used as input to Qlucore Insights. As shown here, we load a BAM file and the Fusion files. And in addition, we also use a patient data file, which collects information about the sample and patient. And we select a model. And here I've selected the BCP ALL model. And then you hit run. And just as an example, I've run this on a sample, which is classified as TCF3 PBX1. We can see in the bar chart that it's classified with a very high certainty, and that the sample is located together with the TCF3 PBX subtype in the PCA plot. We can also see in the Gene Fusion workbench that Ariba and Fusion Catcher detected the TCF3 PBX1 gene fusion. And to the right, we see the results and report preview, where all of this is collected in one final report.